All right, hello everybody. We are going to go ahead and get started on our cell division unit, but let's start by talking about cell reproduction and why cells need to divide. So there's four main reasons. Cells need to replace old cells. Sometimes they need to repair damaged cells, and they need to make more immune cells. So when we're sick, we need to go through a lot of cell division to make our immune system a little bit stronger. We also have to go through cell division in order to make our sperm and egg, which we will talk about a little bit more later. So that's called making our gametes, and that's a different kind of cell division than what we're going to talk about right now. All right, let's just really quickly talk about different forms of DNA. So just like you could have your hair braided or down, the DNA can do kind of the same thing. You can have DNA that looks all messy, and we call it, when it's DNA, we call it chromatin. So when it's all messy like that and it's not all braided up, we call it chromatin. However, when the cell is getting ready to divide, the DNA twists around itself. It's not really braided, but it looks like a letter X, and we call that a bivalent chromosome. So when the DNA wraps up, it condenses and it forms something that's called a bivalent chromosome. So the prefix bi means two, and it has two sides. It has a left side and it has a right side, and they're cinched together with this thing called a centromere. And the left side and the right side are identical copies of each other, and they're called sister chromatids. And whatever's on the left side, it's on the right side also. So if there's a gene on the left side for brown eyes, then on the right side, there's a gene for brown eyes. So that's a bivalent chromosome. Now, the other thing I want to show you, this is a picture of something called a karyotype. This is just a cartoon, but it's a photograph of somebody's DNA and all of their chromosomes lined up side by side. We receive two copies of each chromosome from one copy of each chromosome from each parent. So two copies total. That didn't make any sense. The matching pairs are called homologous chromosomes. So let me say it again. You get one chromosome from dad and you get a matching chromosome from biological mom. And that pair of chromosomes is called homologous chromosomes. The prefix homo means same. They're not the same, but they're similar. So dad's homologous chromosome might carry a gene for brown eyes. Mom's homologous chromosome is also going to carry a gene for eye color, but her gene might be for blue eyes. So the homologous chromosomes, they're similar, but they're not identical. So here's an example right here. The two chromosomes, they look similar in size and shape. They're both bivalent. The one on the left we'll pretend came from the dad, and the one on the right came from the mom. And they have similar genes in similar places on the DNA, but they're not necessarily exactly the same. So one of them might say a smooth chin, and the other one might say a dimple chin, for example. All right, and then we need to go over the terms diploid and haploid. So just like the prefix bi, bi means two, so does the prefix di, di. So a diploid cell is any cell that has pairs of chromosomes from your biological parents. So all cells in the human body are diploid, all cells. So my skin cell here, my chin cell here, my eyeball cell, my butt cheek cell, they are all diploid. That means that they have 46 chromosomes. 23 came from my mom, 23 came from my dad. The only exception to that are the sex cells. Sperm and egg cells have a different name. They're called haploid, and I always emphasize the sound because they carry half the DNA. It helps my students remember. So diploid cells have pairs of chromosomes, 23 from each parent, whereas the sperm and egg only have half the DNA, so they get 23 chromosomes total, so they're haploid. Then when the sperm and the egg come together, they create a baby that is diploid. So if you get half the DNA from one parent and half the DNA from another, and they come together, you have a diploid organism. All right, so now that was sort of the intro. Let's go ahead and talk about what mitosis is. So mitosis is cell division where the two new cells are identical to the original cell. And that's what's happening everywhere in your body except where you're making your sex cells. So not in the testes and not in the ovaries. But everywhere else in the body, if cells are needing to be made, you're needing to reproduce your cells, then it's going to be through mitosis. And it's similar to how bacteria reproduce themselves. It's cloning. So the idea is that you have one cell, it splits into two, and you have two identical cells. So just a couple background pieces of information. The majority of a cell's life is spent in a stage that's called interphase. Interphase, we say the cell is not dividing. So take a look at this cycle. So this is a cycle, that's why it's circular. And the blue section represents interphase. The cell is not dividing. Imagine a female who can get pregnant sometimes in their life, but most of their lives they're not pregnant. That's interphase. They're not dividing. Okay, but then during the yellow stages, that's when the cell is dividing. Some cells enter what's called the G0 phase if they will no longer divide. And this is bad. Our human nervous system cells don't divide. So when we get brain damage or when we sever our spinal column in an accident, those cells don't replace them. So like if you skin your elbow, those cells go through mitosis and you get new elbow cells. But if we have brain damage, those cells don't replace themselves because they don't go through mitosis. All right, so controlling the cell cycle, there's a whole bunch of things we could talk about, but we don't need to say a lot. I just want you to be aware of one thing. There are a set of switches during the cell's lifetime that turn on and off based on whether the cell is growing and dividing correctly. And if there's something wrong with the cell, then the cell cycle should come to a dead stop, and that cell should stop dividing, and then eventually it gets old and dies and your immune system eats it up. That's what should happen. However, sometimes we get mutations in the checkpoints, the things that are supposed to be making sure that our cells are healthy and dividing correctly. Sometimes we get mutations in those. So it says if errors are found, the cell tries to repair itself. So what happens if the checkpoints have mutated, if they fail, and cell division continues when it shouldn't? That, ladies and gentlemen, is what cancer is. So we're going to talk quite a bit about cancer in this unit. But in order to understand cancer, we got to back up and understand how cells divide. So what you're looking at is a series of cells, photographs of basically what should be one cell as it's going through cell division. In this case, we're looking at plant cells, and that's because plant cells are super easy to look at under the microscope. It's a lot trickier to show human cells going through cell division. So what you see is a red color in the middle, and when it's just a solid circle, that's its nucleus and the DNA that's inside of the nucleus. Eventually, the nucleus breaks away, and you see the DNA kind of spreading and separating, and then the cell splits into two. That's mitosis. And the stages of mitosis are called interphase, prophase, metaphase, anaphase, telophase. And everybody says the stupid phrase IPMAT to remember the stages of mitosis. So I will back up and say interphase is not one of the stages of mitosis. That's just like when a woman is in her not pregnant state. Pregnancy starts with prophase. So sort of that's the way it is here. So prophase is the beginning of mitosis. Interphase is just when the cell is doing what it always does, its normal cell life. Then when it's getting ready to divide, that's prophase. Okay, so I'm going to show you. So this is interphase, the cell normal growth and cell activities. The DNA copies itself. The DNA replicates itself. In order for the cell to divide, it has to have two copies of the DNA. Otherwise, when the cell divided, the two new cells would have half the DNA. And then when they divided, they'd have a quarter of the DNA, and we'd start running out of DNA. So in order for a cell to divide, the cell has to copy its DNA. So that happens during interphase. And DNA is all messy, and we say it's in the chromatin form. Now, we're out of interphase, we're pregnant, and we're not pregnant, that's silly, but the cell is getting ready to divide. Okay, so the first stage is called prophase. The nucleus gradually, this is a process, so gradually the nucleus disappears, the DNA winds up. Remember, we learned that it can braid, it's not braids, but it winds up, and it forms what are called bivalent chromosomes. So you see all those little X's, that was the messy hair in the diagram above. Now we see these X's. So the DNA winds up to become bivalent chromosomes, and the centrioles move to the sides. Now, the centrioles are those black and white symbols. All you need to know is that they grow something that are called spindle fibers. And these spindle fibers reach across and they're going to grab the DNA and they're going to push the DNA or pull the DNA to the proper location. But it's just starting right now. So we've had IP. Now we want IP mat. So the next stage is metaphase. And I'll back up too. I will say that prophase looks polka dotty. I use a little bit of alliteration, the letter P and the letter P, to help you when you have to look at a real cell. It's easy to tell on my cartoons what stage they're in, but when you have to look at real cells, it's a little bit trickier. So prophase looks polka dotty. All right, so now metaphase, the chromosomes are lined up in the middle. Let's get my image out of the way here. So it says the spindle fibers, those are the things that are reaching across, attached to the middle of each chromosome, and the chromosomes line up in the middle. So metaphase middle, that's an easy one to remember. The next one is really easy to remember too, anaphase apart. Spindle fibers pull half of each chromosome to opposite sides of the cell. So remember that if you look at the yellow X in metaphase and then it gets separated in anaphase, remember that the left side is identical to the right side. So when we're separating the DNA, we're just going to divide the cell right down the middle, and identical DNA is going to go to the left and the right. So these the chromosomes that look like a letter X, the left and the right side, they're copies of each other. And so the identical DNA is going to go to the opposite sides. So how do you remember anaphase? You say the phrase anaphase apart, AA, anaphase apart. They get pulled apart. And then in the last stage is called telophase. The nuclear membrane comes back and the DNA goes back into its chromatin form, so it doesn't look like a letter X anymore. And then finally, we end with what I always say looks like a big butt. The cell pinches in. Now, this is in an animal cell because animal cells are soft and squishy. Plant cells do it a little bit differently. So when the nucleus has divided, plant cells have to create a new cell wall. So they call it a cell plate. And look at the diagram down below, the green rectangle, and there's a cell plate in between the two new nuclei. So that's what happens in plant cells. All right, so it's our last slide. Can you name the phase? So again, this is a plant cell that's gone through mitosis, and let's see if we can identify. So let's just go through it. It's interphase, that's sort of the normal, the nucleus looks normal. Then there's prophase, and I say prophase, it starts to look polka dotty. That's probably the hardest one. And then anaphase apart. Oh, sorry, I said it backwards. Metaphase middle, the chromosomes are lined up in the middle. And it doesn't look like my diagram because the chromosomes have the consistency of like wet spaghetti noodles. Like they're smooshy. They don't line up perfectly straight. It'd be so nice if they did. So they line up kind of like this. If you were trying to line up spaghetti noodles. So metaphase middle, then anaphase apart. They start pulling apart. That's probably the easiest one to identify. And then telophase, two cells, one telophase. So let's see if you can identify them. I'm going to go ahead and jump in. So the interphase is this one. I wonder if I have it down here. Nope, I thought maybe these went away. Okay, so interphase is this one here. Then prophase polka dotty. Can you see that there's little polka dots of light kind of shining through? That's because the DNA is starting to twist up and form those bivalent chromosomes. And so now light can pass through from the microscope. Light from the microscope can pass through and it looks a little polka dotty. All right, then metaphase middle. Here it is. Here's those spaghetti noodles lined up in the middle. Anaphase apart. They've been pulled apart. The left side of each chromosome has been pulled apart from the right side. And then telophase, two cells. All right, ladies and gentlemen, that's it. Our mitosis lab is next.